Da bare egentlig setter vi I gang. Mine damer og herrer, Dr. Julien Guiraud received his PhD from the Faculty of Medicine of the University of Amsterdam in 2023. He holds a Master of Science degree in Aerospace Engineering from ISAE-SUPAERO. Did I say it right? Yeah. Ladies and gentlemen, say hello to Julien Guiraud.
Thank you. It's wonderful. It's working. So good afternoon. It's a pleasure and it's an honor to be here. I would like to thank the organizer for the invitation. And I'm going to talk about sodium oxybate as a potential substitution therapy for alcohol dependence. But first of all, my declaration of interest. So as you can see, don't trust what I will say. And I will start with a question. Is there a need for substitution therapies for alcohol dependence? Well, you may be aware that the WHO has conducted a study a few years ago analyzing both the burden caused by existing diseases together with analyzing the efficacy of existing intervention to treat those diseases. And based on the result of this analysis, they have established a list of 24 pharmaceutical developments which are a priority. Why? Because they are targeting both high burden disease with an important medical need. And among those 24 priority development, you had the development of new pharmacotherapies for the treatment of cancers, and you had also the development of new treatment for the treatment of alcohol use disorder and alcohol dependence. So it's just to highlight the medical need in this indication. And interestingly, substitution therapies have shown to be quite effective for the treatment of opioid and nicotine dependence. So they may, at least in part, fulfill the current medical need that is observed for the treatment of alcohol dependence. But how could we define a substitution therapy for alcohol dependence? What are the criteria? What are the potential candidates? And what are the data supporting their use as a substitution therapy for alcohol dependence? Well, we try to answer to this question, and we have summarized our findings in the chapter which has been recently published in International Review of Neurobiology. So if you are interested in this topic, I invite you to read the chapter. It's going into much more detail compared to what I will present now. And during this work, we started by trying to identify some proposed criteria for substitution therapy for alcohol dependence. So we conducted a systematic literature search in order to identify those criteria. And we have identified a few of them. Specifically, we identified that a substitution therapy for alcohol dependence should be pharmacologically related to alcohol, that it should be effective both in the treatment of alcohol withdrawal syndrome and in the maintenance of abstinence or in the reduction of alcohol consumption. And finally, we identified that the substitution therapy for alcohol dependence should have a positive benefit risk in this indication. If you look at those criteria, they are very similar to the criteria for substitution therapy for opioid dependence. And for opioid dependence, we already have widely available substitution therapies such as buprenorphine, methadone. So why hasn't a substitution therapy for alcohol dependence already developed and widely available? Well, to try to answer to this question, we need to review a little bit in detail the mechanism of action of opioid and alcohol. In fact, opioids in the brain target very specific receptors, which are called opioid receptors. And they strongly bind to those receptors with a strong agonist action. And this may explain why only low doses of opioid are necessary in order to obtain a strong effect in the brain. In contrast, alcohol is a very different animal. Why? There is no alcohol-specific receptor in the brain. In fact, alcohol is weakly binding to many receptors and quickly binding, it jumps from one receptor to the other, with each time a small action. And this may explain why we need to ingest quite a large quantity of alcohol in order to obtain clinically relevant effect. If we take, for instance, a typical alcohol-dependent subject, he can drink much more than 100 grams of pure alcohol per day. whereas for the treatment of opioid dependence, a few milligrams of methadone are sufficient to treat them. Yeah.
The femhotep-TTE receptors, those are serotonin receptors, right? In the previous slide.
Yeah.
Serotonin.
Serotonin, definitely. Serotonin receptor, GABA receptor, but you will see afterwards I will detail the different action, definitely. And this is the next slide. And in fact, alcohol is interacting with many receptors. It's interacting with an NMDA receptor, producing CNS depressant effect and responsible for withdrawal symptoms. Alcohol is also interacting with the GABA receptors, is a GABA agonist, producing sedation, reduction of anxiety, and its action on the GABA receptor produce indirectly dopamine, creating reward. And other actions have been documented in the literature on several other receptors that I will not go into details here, but they are responsible for subjective effects caused by alcohol consumption. So if I try to summarize, it seems that it's much more easier to find an opioid which will be effective with a positive benefit risk for the treatment of opioid dependence and which will qualify as a substitution therapy as compared to alcohol. Why? Because there is no real specific alcohol receptor. Alcohol is targeting many receptors in the brain, and it's quite difficult to identify a pharmaceutical agent which will exhibit such a broad and specific action on the CNS. And if we look at the existing treatment, they only target some of these systems. This may explain why they have shown only a modest efficacy. So, why not considering ethanol as a substitution therapy for alcohol? Why not? Well, the answer is quite simple and obvious. Unfortunately, ethanol is a highly toxic substance. Why? Because alcohol is metabolized into acetaldehyde, and at high dose, acetaldehyde is very toxic. Why? Because it's responsible for many cell damages, such as DNA damage, immune system dysregulation, and due to these damages, alcohol is associated with over 200 diseases, and it has been ranked as the most harmful addictive substance in the EU. So, unfortunately, ethanol as a substitution therapy for alcohol dependence is likely to have a negative benefit risk, except for a specific subpopulation, specifically homeless individuals with severe alcohol dependence, and literature shows that in this specific population it's very difficult to treat those individuals. And in certain countries, they have implemented an harm reduction approach, which is called the managed alcohol programs, which consists in providing regular doses of alcohol together with health services to homeless individuals with severe alcohol dependence. And several studies have been conducted, and they show benefit for this approach. So this may be a solution for a certain specific population. So, which are the potential candidates? I told you that it's difficult to find a substitution therapy for alcohol dependence. It's difficult, but not impossible. If we recall, a substitution therapy for alcohol dependence should bind to alcohol-sensitive receptors. If we look at the existing treatments, none of them really qualify on this criteria. Why? Because based on their known pharmacological action, they don't really mimic alcohol action on the brain. So, which are the best candidates? Well, to try to answer to this question, we need to identify the substances which bind to alcohol-sensitive sites. And we have the NMDA receptor antagonists, such as ketamine or memantine, which may qualify as substitution therapy for alcohol dependence. We also have GABA receptor agonists, such as sodium oxybate and benzodiazepine. And we have also other compounds. And we reviewed all of them, and the result of our review showed that sodium oxybate is probably the compound which is the closest to a substitution therapy for alcohol dependence. And what is sodium oxybate? Sodium oxybate is a sodium salt of an acid which has a very bad reputation, the GHB. And to understand the history of sodium oxybate, we need to go back to the history of GHB. And for that, we will go back to the 1950s, a period during which a French physician, the Dr. Henri Laborie, wanted to develop a GABA-like which can cross the blood-brain barrier, because a GABA cannot cross the blood-brain barrier. And for that, he used a solvent which is widely available and widely used in various industries, the GBL, the gamma-butyrolactone, and he added an hydroxyl group, and he obtained what we call now the GHB, the gamma-hydroxybutyric acid. A few years later, it was found that GHB was much older than that. Why? Because we all have GHB in our brain. GHB is in fact an endogenous substance which is synthesized from the GABA. And we also found that we can synthesize GHB differently. If you drink GBL, GBL will be transformed into GHB in your blood. This is very important for the safety profile that I will explain later. So remember this. And GHB has shown to have specific properties with alcohol-mimicking action, sedative effect, and given those properties, it has been investigated in various indications. And the sodium salt of GHB has been first tested and approved as an anesthetic agent at high doses in several European countries in the 60s. And then it was approved in the 90s for the treatment of alcohol withdrawal syndrome and the maintenance of abstinence in 2 European countries, Italy and Austria, at low doses. And finally, in the years 2000, it was approved for the treatment of narcolepsy with cataplexy in Europe and in the US and other countries at moderate doses. In the meantime, GHB was also authorized as a food supplement in the 80s and in the 90s. However, due to cases of abuse, misuse reported, now it is a controlled substance and only pharmaceutical sodium oxybate can be legally available. And I'm going to present you the efficacy data of sodium oxybate in the treatment of alcohol withdrawal syndrome and the maintenance of abstinence. And sodium oxybate efficacy in the treatment of and physical dependence with illicit GHB has been reported at doses from 18 to 250 grams per day. So the approved posology is far below the threshold for physical dependence. This is very important to understand the safety data that I'm going to present you. And regarding the safety data, a review has been conducted and published in 2020, and the author conducted a systematic literature search, and they identified and included in their analysis 46 clinical trials investigating the safety of sodium oxybate for the treatment of alcohol dependence, including a bit more than 3,000 patients treated. They also had access to the pharmacovigilance database of sodium oxybate in Italy and Austria, with a bit less than 300,000 patients treated. And what they found is that they found that sodium oxybate was very well tolerated. Well, the most frequent adverse event was dizziness, and it disappeared after the first doses. No death attributable to the treatment has been reported in clinical trial and in pharmacovigilance, and the incidence of adverse events was very low, both in clinical trial and in pharmacovigilance. And finally, the cases of abuse, misuse, dependence, overdose were rare. A higher incidence of abuse was observed in patients with cocaine addiction and alcohol dependence, and also in patients with severe psychiatric comorbidities, but no safety concern was reported. There was just increasing the dose, so we don't know whether they're increasing the dose to have a sufficient effect or whether it was a real case of abuse. In no case reported of drug-facilitated sexual assault. So, in terms of benefit-risk, sodium oxybate have shown evidence of efficacy in the treatment of alcohol withdrawal syndrome and in the maintenance of abstinence, based on positive results from phase 3 trial and pilot randomized control trials, and no safety concern have been reported. So, basically, it is approved and used in 2 European countries under the trademark Alcover, And the European Medicine Agency is reviewing its benefit risk on a yearly basis. And as you can see that the assessments are always positive. So given this positive result and this positive assessment, we decided to try to register sodium oxybate outside Italy and Austria. And we have conducted and we have initiated 2 registration procedures under the name Obvious. And I can tell you it was a nightmare, a true nightmare. I was strongly involved in them. So first registration procedure was assessed by EMA in 2017. And after reviewing the data, EMA came back with a negative opinion, mentioning that for them, the efficacy was not sufficiently substantiated. We were very surprised with this opinion because, first of all, the product is already approved and reviewed by EMA with positive opinion. And secondly, the limitation identified by EMA were reviewed and discussed in the literature, and none of the limitations identified were considered to be really major. So we asked for a re-examination of this opinion. And during the re-examination process, the law allows you to beneficiate, as an applicant, allows you to beneficiate from the opinion of what we call a scientific advisory group. Scientific advisory groups are groups established by the European Medicine Agency. You have one group in oncology, one group in psychiatry, one group in neurology, and these groups are composed of experts in their field, independent from EMA. And they are also they are experts with no conflict of interest with the pharmaceutical industry. And their mandate is to provide independent opinion to EMA. And in case of re-examination procedure, to provide, when the EMA believes that the drug has a negative benefit risk, to have another opinion from experts in the field. So we ask for the consultation of this ad hoc expert group, of this scientific advisory group on psychiatry. The EMA refused. Instead, they convened an ad hoc expert group which provided exactly the same opinion as EMA. And finally, the negative decision was confirmed. We were at that time requesting an unconditional marketing authorization. So a few months later, we have reapplied, but this time for a conditional marketing authorization. And what's happened is that we had exactly the same opinion at the end of the procedure, with efficacy not sufficiently substantiated. So we again asked for the consultation of the scientific advisory group on psychiatry. Again, the EMA refused, and instead consulted ad hoc expert groups. And the ad hoc expert group provided exactly the same opinion as the EMA. So the negative opinion was confirmed in 2020, but at that time we realized that something went wrong, especially with the ad hoc expert groups. And what we found that there was potential violation of the law and of the rules of procedure of EMA. And therefore, a legal action was initiated. And in March 2024, the European Court of Justice, based on the review of the case, provided its judgment and decided to cancel the negative opinion from EMA due to major violation of the law and of the rules of procedure from EMA. This is extremely rare. This is extremely rare. And what's happened? Well, you remember, I told you that we were requesting a scientific advisory group on psychiatry. The EMA has convened 3 expert groups for Obvious. And as you can see, each time it was an ad hoc expert group. And the Court of Justice, the European Court of Justice, I quote, convene an ad hoc expert group instead of the SAG constitute a defect vitiating the procedure for adopting the opinion of the EMA. And if you remember, the objective of the scientific advisory group is to provide an opinion independent from the EMA. And when we look at the composition of the ad hoc expert groups, what we found is that there was plenty of EMA members inside. They were chairing the groups, and there were prominent EMA members, chair of the CHMP, vice chair of the CHMP, head of departments. Well, basically a lot of EMA people were inside the groups. And what we identified afterwards, we also identified that other members, other experts, there were 2 experts working for company developing treatment for alcohol dependence, for competitors. And last but not least, we found that there was 1 expert having his research funded by alcohol producers. That's true. Huh? It's just facts. So basically, the European Court decided to cancel the negative opinion. And now, if you are interested, I can afterwards tell you where we are, because that's another story. We will need 10 minutes, but anyway. So to conclude, sodium oxybate is probably a potential substitution therapy for alcohol dependence. Its mechanism of action is pharmacologically related to alcohol. It has shown evidence of efficacy in the treatment of alcohol withdrawal syndrome and in the maintenance of abstinence based on positive results from phase 3 study and other trials. No safety concerns have been reported over more than 30 years of therapeutic use in 2 European countries. And we believe that, substitution therapy can help patients and can help to fulfill, at least in part, the current medical need in this indication. And I think I am in time. Thank you. Thank you.